This is From the Lab Lectures Part 2, the evidence that the COVID virus came from a lab in Wuhan. Lab leak is not a crazy idea. Lab leaks can and do happen. There are actually several examples of laboratory-acquired SARS infections occurring in Taiwan, Singapore, and Beijing. And there appear to be pretty compelling reasons to believe that the Beijing ones were not handled with the utmost transparency. So this is not a country with a great track record of biosafety risk management or openness in communication. A great amount of work was done by the U.S. government investigation committees to meticulously list specific safety concerns at the BSL-4 outside of Wuhan. A 2018 State Department report gets cited a lot and notes the shortage of appropriately trained staff at that site. Investigators are also doing a lot of sorting through patents and finding things like patents for maintaining better seals on doors, making better liquid disinfectants, patents for improving the design of autoclaves for sterilization. And the point is, these patents kind of inherently make you wonder, why do we need to improve the autoclave? What is currently wrong with the autoclave in the high containment facility? Here's a map of the locations of interest. The Wuhan market is here. The three famous whistleblowing doctors work at these two hospitals, and then the BSL-4 is located here. It's a different campus from the main Wuhan Institute of Virology in the center of Wuhan. There's confirmed work with bat blood samples under BSL-2 conditions here. And the U.S. government's focus on safety concerns is here. So whether your theory rests on escape from the BSL-4 or the main campus of the WIV, biosafety concerns are substantiated. Let's be clear, this could happen. The lab at the center of the speculation is that of Dr. Zhengli Shi. She's a professor at the Wuhan Institute of Virology and has worked there for over 20 years. I'm going to explain the history of coronavirus research and the large part she played in it, and then I'm going to detail the main arguments for this lab specifically being the origin of COVID. Prior to the outbreak of SARS in late 2002, people had no idea that coronaviruses posed a danger to humans. They caused common colds. They were basically harmless. SARS began in the Guangdong province, and fortunately, it was mostly limited to China, though the outbreak did affect Hong Kong and Canada. There were ultimately about 8,000 cases and 800 deaths. The virus had an overall fatality rate of 11%, but 50% in the elderly. The main symptoms were fever, dry cough, and difficulty breathing. I like this Time magazine cover because it captures the sensationalism, but also the naive and insulated perspective of the West at this time. And it reminds us that criticism of China's transparency in handling these types of situations is not new. Sequencing viral genomes was expensive but possible in 2003, so it was easily understood that SARS was a coronavirus, but people were just getting sick. No one knew where SARS came from. In the study of virology, there's this concept called the reservoir, which is the animal with the most ancient infection of a particular class of virus. The reservoir has the longest standing relationship with that virus. For example, the reservoir harboring influenza virus is waterfowl. Humans have been on the receiving end of spillovers from the reservoir into our species many times in history, but the number of strains of flu to ever infect humans pales in comparison to what lies in the reservoir in birds. What we see from observing from our seat in modern times is that the reservoir species has the most strains or the most genetic diversity, and it got that way by millions of years worth of animal and virus evolving together. When animals come in contact with each other, a virus can find itself in a new species. This is called a spillover or a splash from the metaphorically liquid reservoir. Most of the time, spillover events die out. The virus finds itself in a non-native host and is unable to replicate well for a variety of possible reasons, like receptor mismatches or the immune system. Another explanation is that the first transmission may be bloodborne, but maybe the virus doesn't acquire the ability to transmit by aerosol route. Maybe one or two people get infected, but it doesn't really spread. This happens regularly with avian flus in bird markets. Occasionally, when just the right conditions occur, a spillover event is successful and a new host passes it within its own species. From person to person, dog to dog, rat to rat. The chain of transmission may die out in a few months, or it may go on for hundreds, thousands, or even millions of years. So what was the reservoir for SARS? there was a clue, at least, to the proximal origin. Among the first SARS cases were a clear overrepresentation of food and animal handlers. In particular, there were a lot of chefs. This led to the hypothesis that SARS may have come from an animal that people were eating. Each of these chefs was likely getting fresh bloody meat, and each was likely an index case of viral spillover. Sequencing was not good enough and not cheap enough at the time to do a careful analysis of how many independent animal-to-human spillovers occurred, but we know that there was at least 5 jumps because it arose simultaneously out of thin air in different areas of Guangdong in chefs that had no history of contact with each other. Upon learning the occupations of the earliest cases, one scientific group had the idea to sample animals at a market where a chef might go to get meat. They went to the Dongmen market in Shenzhen and found 3 palm civets and 1 raccoon dog that were positive by PCR, meaning they had active infections right then. The group also found antibodies in 2 badgers, which is indicative of past infection. They published these findings in October of 2003. On a map, the Dongmen market is here, pretty far from the first known case, a man in the heart of the city of Guangzhou. With a solid lead on the proximal origin of SARS, the Chinese government tried to stomp out any potential new sources and ordered the culling of many animals at these markets across the province. 2 separate research groups sampled animals at the Jinyuan market at this time. The first found evidence of active SARS infection in 91 of 91 palm civets and 15 of 15 raccoon dogs. This is a 2-hour drive away and over 6 months in time removed from the animals found with active infection in the Dongmen market. The second group found evidence of past or present SARS infection in 14 of 18 palm civets. They tested by both antibodies and PCR. And both of these groups wanted to understand where all of these animals in the market got the virus. The first tested over 1,000 samples from 25 palm civet farms in 12 provinces of China. They found 0 SARS antibodies. The second tested 38 animals on 4 farms. They did find 4 palm civets that were positive for antibodies on 1 farm, but apparently those animals had recently been purchased from a market. So it seems that animals were acquiring infections as they entered the wildlife trade or as they arrived at the markets. The infections were being sustained for months in the markets. How can this be? How can there be active infection all the time, whatever animals are in the markets? The one-time occurrence is from a bat or other animal, probably happens somewhere in the wildlife trade operations. But over the next week at the distribution warehouse, that one animal gives his disease to all the palm civets in his cage, and then they are distributed at 5 markets across the city. If that was the end of the story, those animals would just get over their infection and have antibodies and be immune going forward. But if a fresh crop of naive host is brought in weekly, the way that a market would presumably work, these infections can hypothetically be sustained forever. So just as one group of animals is getting over it, another group is brought in and catches the infection. Here's a strange observation. After all of that work, 1,000 negative test results, there is no single record of a test of a raccoon dog, though those animals are also farmed in large groups for their fur and were also positive for SARS in the markets. This virus popped out of nowhere, killed 800 people. They sampled thousands of palm civets and they didn't test 1 raccoon dog, or at least they didn't report that they tested 1 raccoon dog. Except that the fur industry seems to be very secretive due to a history of being targeted by animal rights groups. I clipped these images from a YouTube video entitled Raccoon Dog Fur Farms in China Exposed by Animal Equality. It was posted in 2015. The makers of this video infiltrated a raccoon dog farm in Shandong, China, and filmed the crowded and filthy living conditions of the animals, as well as the disturbingly inhumane way they are skinned alive for their coats. The voiceover says, "Those whose fur is not in good enough condition are sold to be eaten at this market, where we are not allowed to film the conditions the animals live in. They are finally sold in markets like this one in Wuhan, where the raccoon dogs will die as soon as the client demands." Shandong is not geographically close to Wuhan. It's quite a bit further north. It's a 9-hour drive. Why, for a 3-minute video, do they film in both Shandong and also the Wuhan market, as opposed to any other wet market where wildlife are sold to be eaten? I do not know what to make of this, but it really makes me question how much of a coincidence it really is that SARS-CoV-2 emerged in Wuhan. So fairly or unfairly, the palm civet instead of the raccoon dog took all the heat for being the proximal host of SARS. But palm civets are not the natural reservoir. No one could find a non-market palm civet with the infection. It wasn't until 2 years later in 2005 that the world finds out it's bats. This is where Zhang Lishi comes in. Zhang Lishi and her now longtime collaborator Lingfu Wang found SARS-like viruses in bats. Lingfu Wang is a Duke professor. By 2004, he had a legacy of discovering that horrible new human viruses had their origins in bats. He did this for Hendra virus in the outbreaks in Australia in 1994. The reservoir is bats. It went through a horse proximal origin and then to humans. This pattern is common, the need for an intermediate or proximal host rather than straight bat human. He found a bat origin for Nipah viruses in the outbreak in Malaysia in 1998. Those went bat, pig, human. Bats are flying mammals. They roost in large groups. They're a pretty ideal host for a virus. Lingfu Wang was surely thinking, I bet it comes from a bat. Early in the 2020 pandemic, Zhang Lishi was interviewed by Scientific American, and she described the story of finding the SARS reservoir with Lingfu Wang back in 2004. They spent 8 months traveling around China, climbing through caves, sampling bats. According to this story, they had to be led up steep terrain by village guides. They had to suck in their stomachs and squeeze through little cracks. And every single sample came up negative by PCR. At one point, they were just completely dejected, and they thought that bats may have nothing to do with coronaviruses. But then they tried searching for antibodies instead with a SARS test kit. They'd been searching for active infection by PCR without success. Then they found that all of these bats had antibodies. They were crawling with evidence of past coronavirus infections. The infections are just temporary and seasonal. It's like they've been sampling for flu in Texas in June. It's not a high chance of catching anyone positive. The two scientists and other collaborators published these findings in 2005, and fruit bats were thereafter understood to be the reservoir of coronaviruses. Over the next several years, Zhang Lishi built her entire career off of this work. She and her team traveled all around China to sample bats, sequence coronaviruses, and study their properties. The mission statement of her career can be summarized as the pursuit of two goals. Number one, characterize the reservoir. Where geographically did SARS come from? How many strains are there and how many are dangerous? Number two, determine what precisely allows SARS or other coronaviruses to jump to humans. What properties make it a risk to jump? What aspects of the SARS spike protein make it good at binding human receptors? While Zhang Lishi collected coronaviruses from all over China, she found that southern China, Guangdong in particular, are where most of the strains are harbored. That's where most of the diversity is. So bats fly all around and kick off new trains of transmission, but the infections are most ancient here. This is a figure from Peter Dajak's update on his NIH grant. But further work from other groups has recently extended the hot zone of diversity to northern Thailand, Laos, and Cambodia. So perhaps unsurprisingly, the coronavirus diversity is very large, pretty close to the location where SARS arose. If the vast majority of strains in bats are harmless, having more strains around means more opportunity to come across a harmful strain. In 2016, Zhang Lishi found the closest single sequence to SARS that had ever been identified. It was 96% similar by nucleotide sequence, which is about 40 years of evolution away from the actual SARS strain. She named it WIV16 for Wuhan Institute of Virology 16, and she found the sequence in the Yunnan province of southern China. In 2017, she got even warmer at a remote cave also in the Yunnan province near the town of Kunming. She didn't find the SARS sequence exactly, but she found all the pieces that could form the SARS strain via recombination. The bats in this single location harbored the strains of coronaviruses that between them contained all the genetic components of SARS. And this is like as close as you're ever going to get. Finding this cave was kind of the crowning jewel achievement at the time and earned her international recognition. Only finding SARS in bits and pieces is not unexpected because there are no original coronavirus strains. They are all recombinations of each other. In the short term, you can observe a set of viral strains acquiring mutations and becoming more distantly related over time. Common ancestor, progeny acquire mutations, become more divergent over time. But at some point, one unlucky bat gets 2 infections at once. And when a cell in the bat is co-infected with 2 coronaviruses, their genomes kind of get randomly shuffled, and we call this recombination. There's a few consequences of the propensity of coronaviruses to mix genomes frequently. It becomes non-trivial to say what virus is most closely related to another virus because the answer will be different in different parts of the genome. It is not uncommon to find viruses that are 90% similar in one portion and under 20% similar in another portion. So unfortunately, it becomes incredibly complicated to map out the evolutionary history cleanly because the deck of cards keeps getting reshuffled and everything is so profoundly mixed up. Coronavirus researchers picked a portion for classification just because they have to pick something. They settled on the RNA-dependent RNA polymerase gene, which is the first thing that Zhang Lishi sequences in every one of her samples. It's kind of like getting a fingerprint. And it provides a lot of utility to assess the relatedness due to mutations arising one by one over time, but with a known caveat that it could be a poor predictor of relatedness in other parts of the genome. Zhang Lishi, Lingfo Wang, and Peter Daszak got the idea to test villagers who live near that cave where she found the different flavors of SARS. In 2018, they published that 3% of people in these villages had antibodies to SARS-like coronaviruses, though they had no recollection of interaction with the bats. A similar later study combined that SARS antibody status with a survey on occupation, and that was done in Malaysia, and it suggests that hunting and mining is strongly associated with acquiring a SARS-like infection. 33% of miners and nearly 40% of bat hunters had antibodies in a pre-COVID world. So the thought is that viral spillovers are happening all the time, but it's usually just the hunter, just the wife that get infected and it dies out. This isn't really unexpected if we think about other human pathogens that arose from cross-species transmissions. HIV has crossed to humans at least 12 times, but only one is the HIV that you know about, the strain that amplified in the growing city of Kinshasa in the 1960s. Ebola crossed from bats to humans at least 19 times before it reached a city and resulted in the harrowing outbreak of 2014 in Sierra Leone. So it takes a city. Even for the most infectious virus, there has to be enough contacts, there has to be enough opportunity for transmission, there needs to be a few super spreading events, or the math just doesn't math and the outbreak dies out. Rewinding a little, except for Zhang Lishi and her colleagues, after SARS, the world essentially forgot about coronaviruses for a decade until the first outbreak of MERS in Saudi Arabia. MERS is Middle Eastern Respiratory Syndrome, and it has the fever, cough, and shortness of breath symptoms that seem common to these infections, but it was more deadly. The first outbreak had a 50% fatality rate, and it was discovered that MERS is harbored by camels. Bats are still the ultimate reservoir, but camels were the proximal origin. Whoever had reason to be studying the camels had their blood banked back to 1992 and was able to show that they did have antibodies even at that time. So why MERS never arose in people prior to 2012, we don't really know. It could be that there's some kind of change, like practices with camel meat consumption, or it could be that only in modern times do we investigate when a lone herder dies. It isn't really known. Instead of one striking outbreak and dieback like SARS, though, MERS only infects a few people at a time, usually an index case, very close contacts, hospital workers. There's one major outbreak that sickened over 100 people, but for the most part, MERS is just this persistent, terrifying irritant that keeps popping up, killing a few people, and disappearing. This happened from 2012 onward, at least until COVID-19, which provides some cross-immunity. Zhang Lishi didn't play much part in the study of MERS outbreaks, but she did in the next harmful coronavirus spillover. Besides SARS and MERS, and of course SARS-CoV-2, there is one other modern-era coronavirus of note. It's called Swine Acute Diarrhea Syndrome, or SADS. In 2017, SADS killed 24,000 pigs. It arose in Guangdong, so same location as the SARS outbreak. Veterinarians actually called in Zhang Lishi to investigate. Her team took samples, sequenced them, discovered a coronavirus, identified the closest sequence in her database, isolated the virus, and tested whether it could replicate in various types of cells. They found that it could not use human ACE2 to enter cells. And this was all a real success story. It hammered home the clear and present danger status of coronaviruses in the region, even if this one was not that dangerous to humans. It was a risk to commerce and the food supply. And it occurred just 2 years before COVID, and it kind of gave her a dry run in outbreak response. She ends up doing essentially the same series of experiments in 2020. So here we are, January 2020, very early in the outbreak in Wuhan. Zheng Lixi published a paper about the novel coronavirus. It was published February 3rd, but it was received by Nature on January 20th. She acquired patient samples from the Jinyintan hospital where the earliest cases of COVID had been transferred. According to her timeline, she got a call from her supervisor while at a conference in Shanghai on December 30th, and she took a high-speed train home to Wuhan. So apparently she did all of this experimental work and authored the paper in 3 weeks. And I would comment to say this is feasible, but it would mean dropping everything in your personal and professional life for that time. The samples were positive for SARS-like coronaviruses by PCR with multiple primer sets. The hospital doctors had figured out that this was a coronavirus like SARS, but this is the first paper that says the Wuhan outbreak is a coronavirus. There were antibodies towards a SARS-like virus. She isolated the virus, meaning she put sample on permissive cells and coaxed it to start replicating. And with actively replicating virus, she could determine which species ACE2 receptor the virus could use from an array of cell lines in her lab. She got the full-length sequence from 5 of the patients, and she searched her database to find the closest bat coronavirus, which was RATG13, and I'm going to call it RATG13 for simplicity. And it was 96% identical. The full-length RATG13 sequence had not been published anywhere before COVID. RATG13 is this blue line here. This is a graph of the percentage similarity to the pandemic strain. The closer it stays up towards 100, the more similar. And she's showing several other bat coronaviruses of interest for comparison. And they all dip down, even RATG13, a little bit in the spike portion of the genome, because that's the fastest evolving and most diverse part of the genome. But for the most part, RATG13 is pretty close to SARS-CoV-2. RATG13 became controversial. 2 scientists noticed something. These 2 are a couple, and they appear to work in biotech in India. They are Dr. Monali Rahalkar and Dr. Rahul Bahulekar. What they noticed is that there was a short sequence, about 400 base pairs, in the GenBank database that was a 100% match to RATG13. That sequence was an RDRP fragment deposited by Zheng Lixi's lab. The sample that matched RATG13 was called ID4991 and was associated with a paper of Zheng Lixi's from 2016. Coexistence of multiple coronaviruses in several bat colonies in an abandoned mine shaft. The paper describes a mine shaft in Mojang County in the Yunnan province where bats were sampled from 2012 to 2015. The couple also read Zheng Lixi's interview with Scientific American, which was originally posted online March 11th. In the interview, Zheng Lixi discussed being called to investigate pneumonia-like diseases in 6 miners in 2012. She talked about the sampling that she did in the following years and the diverse group of coronaviruses that she found there. Then Rahalkar and Bahulekar found a news article from Science magazine called A New Killer Virus in China. The article is about 6 miners who fell ill with pneumonia. They were clearing bat guano and fungus from an abandoned mine in Mojang. 3 of them died. The article is not about Zheng Lixi's team or coronaviruses, though. It's about another viral research team that found a novel paramyxovirus in local rodents. This group seemed to be able to drum up the glory. They named their virus Mojang paramyxovirus, and they got to do an interview with Science magazine, but they were never able to actually prove that the miner's illness was attributable to that novel paramyxovirus. So Rahalkar and Bahulekar are irritated because they have deduced that RATG13, the closest virus to COVID-19, was sampled from a mine shaft where people died of pneumonia after exposure. And they believe that Zheng Lixi may be trying to obfuscate this fact by not citing the prior publication and not giving a detailed explanation of RATG13's sampling location. The Zheng Lixi paper only says, we found a short fragment which was previously detected in a sample from Yunnan province, showed high sequence identity to SARS-CoV-2. Rahalkar and Bahulkar wrote a paper connecting these dots, and after the preprint was published online, they received an email from someone calling themselves the Seeker. The Seeker had identified some documents that bolstered and corroborated the story that they had already pieced together. And the Seeker is a member of Drastic Research. Drastic stands for Decentralized Radical Autonomous Search Team Investigating COVID-19. On the team are both anonymous and named members, the Seeker being a prominent contributor. The Seeker provided the authors with a master's thesis and a PhD thesis found on cnki.net. This is the Chinese server that houses these kind of documents, so this is legit. The master's thesis details the cases of 4 of the 6 men, what they were doing in that mine, as well as their symptoms, their treatments, some lab results, x-rays, and CT scans. Some of the described symptoms are arguably similar to COVID-19, with fever, cough, and shortness of breath being common between the cases. It also says that the Wuhan Institute of Virology tested at least 1 patient sample, and the serum was positive by IgM for coronaviruses. There's no data to look at. It's simply an account according to a presumably in-person conversation. The PhD thesis gave a little bit more information about the testing at the WIV. Kenping Huang was the PhD student at the Chinese Centers for Disease Control and Prevention in Beijing. His research work was to go back to that mine, sample bats, sample other rodents, other wildlife, take it back to the Beijing lab and look for viruses. But in the background and discussion portions of his thesis, to explain why he's doing what he's doing, he says that the WIV tested blood samples and throat swabs from those miners. They were negative by PCR for SARS coronaviruses, but blood samples were positive for SARS-like antibodies, specifically IgG antibodies, a detail which does differ from the master's thesis. Again, this is a no data statement, which is kind of unusual for this type of document. Regardless of their divinity, these theses add concerning extensions to the Rahalkar and Bahulkar point. Rachi 13 came from a mine in Mojang for sure, but further, the WIV and the Chinese CDC in Beijing had reason to argue or believe that the miners may have had a coronavirus. So to summarize, Zhang Lixi changed the name of the sample from ID 4991 to Rachi 13, failed to cite the prior publication of the small piece of sequence, and did not explain precisely where it was sampled. And she, didn't say that this was a mine where 3 people died from mysterious pneumonia. Here's the counter argument for why this is not that weird. She says she sequenced a short RDRP fragment previously. There is one 100% match RDRP fragment in GenBank, and it was deposited by her. How close can dots be before it doesn't count as connecting them anymore? She said it was sampled in the Yunnan province. The name itself contains a more precise sampling location. It is the species of bat, Rhinolophus affinis, sampling location, Tongguan, and year of sampling, 2013. Tongguan is in the Yunnan province, where she said it was sampled. That is accurate information. And that 4-digit code, that 4991, that's a She Lab naming convention. Rachi 13 is a field naming convention. It's more formal and appropriate for the publication. So, of course, she used a more formal name for this Nature paper. And because all the miner samples tested negative by PCR, she really doesn't have any proof that those miners had coronaviruses. Antibody results alone are not conclusive. Negative by PCR is a big blow. To illustrate, the miner's illness was not mentioned as a reason for her interest in the mine in the first publication either. So that was a much less controversial situation. Overall, the information brought to light in this paper is accurate, but we're doing a lot of scrutinizing of minutia and speculating on intent. Whether the symptoms described are truly consistent with coronavirus infection is debated. The primary hallmark symptoms of SARS, MERS, and COVID are fever, dry cough, and shortness of breath. These miners had fevers, coughs, and difficulty breathing. But this group, in their own words, propose a more balanced interpretation of the case studies and highlight what they claim are major discrepancies with COVID-19. I'm not a physician, but the most understandable discrepancy is that the miners had wet coughs. not dry coughs. They were coughing up blood, and they were coughing up different colored sputum. The tangentially involved drastic team continued to pick at anything they deemed strange about rat G13, and they did so in a way that was aggressive and prolific. I'll walk through a handful of examples. They find what they believe is an inconsistency between the text of the paper and the file names of some of the raw sequencing reads of rat G13. These two sentences in the paper make it sound like Zhang Lixi's team sequenced the fingerprint in 2016, became aware of COVID, went to the freezer, and sequenced the rest of the genome specifically because there was this outbreak. But they actually sequenced rat G13 far earlier. There are raw sequence files that they uploaded to publicly accessible databases that include a date as part of the file name, and some of those dates are 2017 and 2018, pretty clearly suggesting that those sequencing reactions were performed prior to the outbreak. One could interpret this as the lab not being transparent that they were working with at least the RNA in the intervening years, or one could interpret this as Zhang Lixi choosing her words carefully to be accurate but succinct. It would be unconventional to detail out a needlessly complex story to explain that some parts were sequenced in 2017, then the rest of the genome in 2018, except for some holes that they filled in after the pandemic. Journals had word limits. That would be a very strange approach, actually. The drastic team also has a problem with the 7896 label. 7896 is another one of these 4-digit isolate serial numbers from Zhang Lixi's lab. It's a virus related to SARS-CoV-2, but not as close as rat G13. And the drastic team speculates that something fishy is going on, like 7896 virus was somehow used instead of rat G13 for these reactions, or even outright accusing the lab of falsifying the data by combining the rat G13 sequence with the 7896 sequence in a computer. They don't provide any rationale for this accusation other than the 7896 number being in this file name. But see these F's and R's. Those stand for forward and reverse. These are primer names. Primers are a critical component of the sequencing reaction. It controls the starting point. The files are named by sample, which is rat G13, primer, date, and the well orientation of a 96 well plate. This could not be more ordinary. Clearly, the primers were designed off of the 7896 sequence at some point in the past, and they used those primers on rat G13. I use primers all the time where the name of the primer comes from something I did 7 years ago and has absolutely no relevance to what I'm using it for now. This is super normal and certainly not evidence that rat G13 is a falsified sequence. The team then identified what they believe are suspicious inconsistencies in timeline and status of the sample. A New York Sunday Times interview with Peter Daszak says, however, after sequencing the full genome for rat G13, the lab sample of the virus disintegrated. The drastic publication angrily mocks the word disintegrated because it's like, okay, a very important sample in your freezer just spontaneously disintegrated? That's very suspicious. But there are not quotes around this statement. I would bet that Peter Daszak told the journalist the sample had degraded. RNA is unstable, degraded being the conventional language to describe an RNA sample becoming unusable after too many freeze thaws or after too much time at room temp. The journalist likely either misheard him or just wrote the word disintegrated and thought it was an equivalent word. Last example, Zhang Lixi uploaded the first 15 nucleotides of the rat G13 sequence much later than the rest of the genome, not until October of 2020. This, in and of itself, is not suspicious because the bitter end of a sequence can be more technically challenging to capture. There are a lot of SARS-like viruses labeled as complete but missing the first 30-ish base pairs. But before Zhang Lixi added that last piece, she was caught in an interview saying she had the whole genome except the first 15 base pairs. And the drastic team argues, how could she get this sequence if the sample had disintegrated? And how could she know that it's missing exactly 15 nucleotides, not 14 or 16? But SARS-CoV-2, related bat viruses, they have 15 nucleotides there when you compare by alignment. It would be strange to assume that rat G13 was missing anything other than precisely 15 nucleotides. So what I think is happening here is that there's a mismatch between theoretically understanding molecular biology from reading and ever having stepped foot in an actual lab. The drastic team is not composed of biologists. There was one virologist, but she left and is now a proponent for natural origin theory. The drastic team is businessmen, security people, IT people, and seemingly hackers doing this in their spare time, and they're lacking a grasp of what life looked like on the ground. They're framing really ordinary behavior as suspicious, and they have just enough detail to sound credible. And if you do enough of that, it starts to seem like there's this overwhelming stack of suspicious activity when really there may be actually very little suspicious activity. But they are good at digging up stuff. There's one more angle to this Mojang mind theory. Drastic member Francisco de Assis, or I'll just call him Francisco, combed through Zhang Lixi's publications and sequence deposits and created a large spreadsheet of sampling dates, locations, isolate serial numbers, and by recording information when it was available, he could deduce and fill in the gaps when it was not, when she must have been traveling where and what samples came from which trips. He also taught himself how to make these phylogenetic trees, and he found that there was a group of 8 coronaviruses relatively close to the SARS-CoV-2 lineage, and based on his spreadsheet, he knew that those samples also probably came from that Mojang mine. Here is SARS-CoV-2, and here is VAT-G13, very close. And here is the lineage in question, one of which is that same 7896 number that was in the file names before that they didn't like. Francisco is doing a lot of tweeting, asking why there has been no additional study of these viruses, which appear to be very close to SARS-CoV-2. After all this accusation, Zhang Lixi published an addendum to her paper in November of 2020, essentially saying, "You are totally right. The series of events is as follows. She received samples from 4 of the 6 miners in 2012. Those samples were negative by PCR, and when testing for presence of antibodies, none of the samples gave a positive result." However, she sampled that cave from 2012 to 2015 once or twice a year. They found 9 SARS-like beta coronaviruses. In 2016, she published a paper and deposited many of the RDRP sequences into the GenBank database, including ID49N1. In 2018, when next-gen sequencing became cheaper, she sequenced the entire genome of a lot of virus samples stored in her freezers, including ID4991, and the IDD491 sample included in the first paper was renamed RATG13 to align with convention in the field. The story adds up. The drastic team had already hypothesized that there were precisely 8 similar sequences in addition to RATG13 taken from the same location, and then she's saying that there's 9 total. But there's still this odd discrepancy between the thesis saying that the samples were positive for antibodies and her saying here that they were not. And I noticed that she says PCR tests were negative and antibody tests were not positive. And I suspected that was carefully worded. I speculate those samples were slightly above background, in the gray zone between stone cold negative and like maybe if you squint. They must have inspired and have hoped to continue to sample the mine, even enough to tell their collaborators some conflicting information over the years, but maybe not enough to have a leg to stand on in the harsh light of peer review. But we are not provided with an explanation of how this discrepancy in stories can be explained. The members of the drastic team were really interested in the 8 other sequences sampled at the mine and were very critical of the lack of urgency in publishing any additional information about them. The thinly veiled accusation is that one of them could actually be the origin of COVID. The timeline is as follows. Zhang Lixi publishes the coronavirus outbreak Nature paper in February. She's then interviewed by Scientific American, and that goes online in March. The Mojang mine theory paper is published in May. Zhang Lixi published the addendum to her paper in November. Then the drastic team attended scientific conferences during late 2020 and throughout 2021 and took pictures of her slides. This talk's from December 2020. She showed a phylogenetic tree, and these are the 8 sequences in question. On this day, they are labeled by RA and then her lab's isolate numbering system. Then in another talk, she labeled them Mojang bat covies, and this is the first absolute confirmation that those sequences came from the mine. And then in yet another talk, after the Mojang mine and other 8 theory was generating some heat and she was fielding questions, she took those labels off of her slides altogether, and they are, of course, very critical of that. But there's something about the Mojang mine and other 8 theory that doesn't make sense. Something big that the drastic team seems to be completely ignoring. In early October of 2019, Zhang Lishi and Peter Daszak submitted a paper to Nature Communications. The paper is about the big picture evolutionary history of bat coronaviruses, the location and dispersion of clades and strains, and how often viruses switch bat species hosts, which appears to be pretty regularly. Yuck. The paper was submitted October 6th of 2019, but then the pandemic happened, so I assume there's a lot of recontextualization and updating needed, and it was finally published in August of 2020. And it included a huge data dump, 630 novel CoV sequences from the WIV, mostly all just that short RDRP fingerprint to provide the framework for figuring out all this evolutionary history, useful or not. This paper is the source of the sequences driving the other 8 speculation. But all of these sequences were submitted to the GenDank database August 13th of 2019. 7909, 7952, 7921, 7924, 7905, 7907, 7931, and 7896. They all say version 1. They were all submitted August 13th of 2019. And these sequences are decidedly different from SARS-CoV-2. The top sequence is Wuhan 1, the SARS-CoV-2 reference, and the second sequence is RATG13. RATG13 is 96% similar to SARS-CoV-2, and you can see how much more similar it looks by eye than to the other 8. There are like 6 differences here compared to twice as many, at least, in the other 8. Those all look very similar to each other and pretty different from SARS-CoV-2. I am confused as to why the drastic team so aggressively pursues the idea that the sequences they highlighted could somehow be related to the origin of COVID-19. They got those sequences from public databases. The submission date predates the pandemic, and those sequences don't match COVID-19. Like, this is very definitive evidence that they're not COVID-19. But the team clearly thinks that these close but no cigar sequences could be turned into SARS-CoV-2. But that's not really how it works. If we just start with RATG13 itself, not even the more distant other 8, you cannot use this as a base and engineer or by chance arrive at something that is later found to exist in nature like Banal 2052. 1 month after the drastic team wrote up their 7896 clade investigation, Zhang Lishi submitted a response paper, which was published in Emerging Microbes and Infections in December of 2021. She published the entire genome of one of the 8 sequences and named it RATG15 and claimed that the other 7 were all collected in the same trip in 2015, and they were all pretty much the same sequence, which is consistent with what we already know from the RDRP fragment. She claims that while the RDRP part of the genome is about 90% similar to SARS-CoV-2, that similarity drops off to 50% in the first half of the genome and even under 30% in the spike region. She specifically entitled the paper to emphasize that these viruses use the bat ACE2 receptor and are not capable of using the human ACE2 receptor. So that would mean that these viruses would fail her first pass test for interesting virus to screen or use in gain-of-function experiments. She then shows a tree of the major lineages of beta coronaviruses with the original SARS lineage in red, SARS-CoV-2 lineage in blue, and the new set of 8 in purple, forming their own distinct clade. And then in blue, along with RATG13 and the pangolin viruses that we've already talked about, there are now a number of new additions since the beginning of the pandemic. Viruses found in Cambodia, Thailand, even Japan are closer to SARS-CoV-2 than the RATG15. These viruses were found by other groups, sequences put in public databases by scientists from multiple different countries, some of them identical in the receptor binding domain to SARS-CoV-2, just like Banal 2052. The drastic team is fixated and aflame with anger that there are no studies on like the 11th most interesting viruses. Without provocation, there was no reason for Zhang Liqi to publish this paper. For better or for worse, the publication process filters for positive results, and these are negative results. Its sole reason for being publishable and of interest is because of the accusation. In the end, let's remember that Zhang Liqi's interview itself catapulted the Mojang mind theory. Neither Drastic nor Rahalkar and Bahulkar put it together until her interview. In a pre-lab leak theory-filled world, Zhang Liqi likely thought she was getting her day in the sun as a researcher who turned out to be right. She'd been trumpeting the clear and present danger of coronavirus spillover, and here we are. Interview me. She was not shy about the miners' story. She said fungus ultimately killed the miners, which apparently was the physician's conclusion. But she also said it would only have been a matter of time before they caught coronaviruses if the mine had not been promptly shut. Obviously, she believed that there were strains in that mine that could have jumped to humans, and she told the first journalist she could find. The Chinese government was censoring scientific discussion. Her slides would be under the most intense scrutiny imaginable. Talking about the Mojang mine sequences at European conferences, waltzing up to Scientific American and talking about these miners, both of these actions would be unthinkably reckless if COVID came from a virus sampled in that mine. There's one other major reason people think that COVID came from this lab. The sequence database at the Wuhan Institute of Virology was removed from public viewing. The database had thousands of entries of viral pathogens. It included bat coronaviruses from Zhang Liqi's work, and it was taken offline September 12th of 2019. The Drastic team got their hands on some site metadata and published the dates, page visits, download requests, and download size during the relevant periods of time. The first version of the sequence database went online in April of 2019, so just 7 months before the estimated index cases. From April until September, this database was getting between 10 and a few hundred visits per day, except in June. On June 15th and 16th, no usage whatsoever. Then on June 18th and 19th, there were about 600,000 visits and 6,000 megabytes downloaded, which is a strange anomaly. The downloader was the China Science and Technology Network, which is basically the internet runners of China. The download size was equivalent to downloading the entire database 10 times. I'm hard-pressed to think of anything you can do with 10 copies of a database that you can't do with one, but I have no idea what happened, nor does the Drastic team. There was an odd period of no usage whatsoever for 2 weeks in August, but on September 12th, the database was taken offline, and there's no usage at all after that for a while. It mysteriously came back online mid-December and was available for 4 days, but no site visits. Then it was online and offline, sporadic and random hours after that. There were no site visits during this period of on and off glitches. The database was taken offline permanently in February of 2020. Every time that Zhang Liqi has been asked why the database was taken offline, she has cited security concerns. She describes thousands of hacking attempts since the start of the pandemic, and the IT managers were worried someone might implant sequences with malicious intent. But she has to be referring to the February instance, where the database was taken down permanently. She's never addressed why it went down in September or why it was sporadically on and offline for December and January. If the database had been online for years, then in late November of 2019, it went dark. I would consider that fairly strong circumstantial evidence of lab leak. If the database had been online for years and then on December 31st of 2019, it went dark, I would also consider that fairly strong circumstantial evidence of lab leak. There's a lot of scrambling and evidence of information transfer around that time. And it is when Zhang Liqi hypothetically got the first patient samples from the Jinyintian hospital. But a fairly new database has a bunch of issues, goes down in September, is intermittently on for December and January, like these key periods of time, then goes down permanently in February after lab leak rumors gain steam. I don't feel that timing strongly correlates with anything interesting. No one can prove a negative. No one can prove that Zhang Lixi's lab was not working on a viral isolate, an unpublished sequence that was COVID. I think depositing the genetic fingerprint of 630 coronaviruses, presumably most of any interest ever sequenced in the lab, submitting that 3 months prior to the start of the pandemic, that's a pretty good alibi. In my mind, there are 3 possible scenarios that remain for COVID-19 to have originated from lab leak. The first scenario requires secretly deleting data from the GenBank database or otherwise spoofing the submission info. Like if COVID-19 was among the 630 sequences, but not published, and they convinced the NIH to take it out. I don't put this out of the realm of possibility, but we do have a lot of FOIA emails, and something like this has yet to be found. The Energy Department did find that a doctor in Beijing tried to upload the sequence on December 28th. For some reason, that's touted as suspicious, but it's not really. The timeline makes complete sense. There was a doctor in Wuhan, Dr. Ai Fen, at the Wuhan Central Hospital, and she sent a sample to an analytical lab called Vision Medicals for sequencing to diagnose a patient in her ER. Vision Medicals called her with the SARS result on December 27th. They did not just get a SARS result and go on with their day. They contacted the Chinese Academy of Medical Sciences, and they were working with them. And somebody that works there, Dr. Li Ren, submitted the sequence to GenBank the next day and labeled it Wuhan coronavirus 2019. There were multiple other doctors working with multiple genomics companies who all have this sequence by late December. The official story is that Dr. Zhang Yongzhen submitted it to GenBank on January 5th. He sequenced it in his lab, submitted it to GenBank January 5th. But by the 10th, it still hadn't gone online. GenBank is not TikTok. One does not simply just post. He instead then emailed it to Edie Holmes, his collaborator, who put it on a virology social media site for speed. And for some reason, people trying to say vaccine development timelines aren't adding up, they use this January 10th date as though everyone in China is helpless until somebody barges through both Chinese gag orders and American bureaucracy and gets the sequence truly public. But a lot of people in China have it by then. All this to say, I would hope but not assume that the Energy Department's competence in finding this aborted submission on December 28th would mean they could also find anything that Zhang Lixi requested to be deleted. A second scenario is that the COVID outbreak started far before November of 2019. Could that somehow make all of this actually align? Could the comprehensive bat coronavirus paper, while an absolutely heroic amount of work, could that be written specifically to be an alibi? I don't think this is a great theory. The sicknesses would have to occur in July or earlier for an August submission. Then, for some reason, they wouldn't take down into the database until September. And everyone's molecular clock analysis dates the origin at late October to late November. And a July origin doesn't really jive very well with the report that 3 WIV lab workers got sick in November. So there's a lot of timelines that just aren't really adding up with that theory. A third scenario is that the COVID virus was the one virus that was never published because it was special. It was secret for some reason. It was an operation for a vaccine or something. Or it was a lab leak, but it had absolutely nothing to do with Zhang Lixi's lab. I think this hypothesis is the most plausible. By that, I mean there's not evidence for it, but it's not actively implausible. But it also means that the allegedly suspicious observations about this lab are no longer relevant to the theory. So all of this detailing of the gain-of-function work, the furin cleavage site, the emails, the DARPA proposal, the Mojang mine, and the database are red herrings. This would be a separate hypothesis and would need its own story. Okay, so all in all, a lot of hard work, no actual evidence of a lab leak, still hypothetically plausible, cannot prove a negative. Check out the next episode to hear what they found at the Huanan market.